Hello friends, myself Dr. Piyush Tailor. I'm Associate Professor in Government Medical College, Surat. Today we are going to learn about the lipoprotein metabolism. In previous two lectures, already we have covered about the introduction about the lipoprotein and apolipoprotein. We have covered up something about the significance of apolipoprotein also. And now we are going to discuss about the metabolism of individual lipoproteins. So in this part of the lecture, we try to learn about the chylomicron metabolism. Now, we already understood that chylomicron gets synthesized into the intestinal mucosa. Okay, when we take any amount of the fat from the food, fat in a food, then ultimately that food will go to the intestinal wall, and the intestinal wall again it get re-esterified. When it get re-esterified, then all the re-esterified fatty acid, cholesterol, which is converted to triglyceride, phospholipid, will combine together and form the one other big molecule, that is chylomicron. Now we already know this chylomicron having the lowest density amongst all the lipoproteins. Reason is that it having the highest amount of triglyceride. So we know that when it's formed into the intestinal mucosa, then it is not going to the blood vessels. It will go to the circulation to the lymphatic system. It will first of all absorbed by the lymphatic system, and then after through the lymphatic system it reaches to the liver. As already discussed, there is a reach in triglyceride, and when it combined into intestinal mucosa, they having some specific apoproteins, that is ApoA and ApoB48. Now when we discuss about the ApoA and ApoB48, at that time we come to remember that what exact function is done by these lipoproteins. In the blood, when it transported in intestine to other tissue, then some other apolipoprotein also added, that is ApoC2 and ApoE. So practically speaking, we can say when the chylomicron is formed and when it reaches to the circulation, it having the ApoA, ApoB48, ApoC2 and ApoE is there, and these two will be added to the SDM. In next figure, we discuss again this part, and then you can understand in a better way how the lipoproteins are added with the chylomicron. Now this is also you have to remember as a clinical purpose, that if the lipid serum is kept overnight in refrigerator, chylomicron rises as a creep layer on top. As already understood, it's having the lowest density. So when you keep in a refrigerator, when the serum gets separated, in the serum also the chylomicron comes on a upper layer. So this already you know about this figure. And when in intestine and bile salt act on a lipid molecule, that will be emulsified. Once the emulsification will develop, there will be a micelle formation is there, and that micelle will help for the absorption of all that lipids which comes through the food. And when all the lipid with the protein, when combined together form the chylomicron, and that chylomicron will go in the lymphatic ducts. Okay, and through the lymphatic duct, first of all it will reach to the blood, and then after it will reach to the liver. Now, here just we are taking a brief of the metabolism related to chylomicron. Here you can understand when we are taking the dietary triglyceride, will come first of all in the small intestine, then in the lymphatics. And lymphatics, when the chylomicron get formed, as we discussed, having the ApoE is there and ApoB48 is there, and in the core there is a triglyceride and cholesterol is there. This we call the chylomicron. But here it is not a mature chylomicron. Later on, some other lipoprotein also get added, and then after mature chylomicron get formed. So here you can see the difference between this chylomicron, this micromicron is that here ApoA and ApoE is added. Now it comes to the HDL, which is there in the peripheral blood. Okay, so this HDL, when it goes to the breakdown, that will donate some other apoprotein like ApoA and ApoE, and when finally the chylomicron get formed, it having the 4 lipoprotein, ApoA, Apo48, ApoE and ApoC is there. Now already we have understood that all the lipoprotein having the different function. So also we just think about the ApoC. As we have discussed, this is a apolipoprotein C2 is there, and that C2 having the main function or activation of lipoprotein lipase enzyme. And because of the activation of this enzyme, whatever the triglyceride is there or cholesterol ester is there, it's get broken down. And because of the breakdown, we know that when it's broken down, glycerol get formed and fatty acid get formed. Whatever the fatty acid is there, it will be utilized by the extrahepatic tissue. You may say it may be utilized by the muscle for the energy purpose. And glycerol may go to some other gluconeogenesis process. It might be used for the glucose formation also. It may be used for the energy purpose also. After this process, whatever the molecules remain, that will be the chylomicron remnant. But in this remnant, it having only B48 and E is there. Now it doesn't having the A and C. It's having the B48 and E is there with that molecule. Then you got the chylomicron remnant. These two molecules will help. These two molecules will help the chylomicron to get internalized into the liver. Here, ApoE will act as a ligand, and through that ligand, that chylomicron will bind with the receptor which is attached on a liver, and it get internalized, and thereafter the further metabolism will get happen with the chylomicron. So we are just taking a brief of the same thing. As you know that it mainly happen in the adipose tissue and skeletal muscle. Half-life for the chylomicron is very low. It is only up to the 1 hour. So you can say when we take the food, then when the chylomicron gets generated, and that generated chylomicron remains in the blood only for 1.5 to 2 hours. If half-life is 1 hour, and maximum of the chylomicron will remain for the 1 hour. And that is why immediately after the food, we avoid to take the sample, because blood sample for the blood testing. If you take that sample, that might happen that blood might be having the high amount of chylomicron, and it may interfere with the analysis. Same way that this endothelial layer of the capillaries of the adipose tissue as well as muscle and heart having the lipoprotein lipase enzyme. All these three, adipose tissue, muscle and heart, adipose tissue, muscle and heart, all the three having the lipoprotein lipase enzyme that we already understood, it is activated by the ApoC2. Once it get activated, we already understood it converted to fatty acid plus glycerol, and that will be utilized for the energy purpose. Another significant important things about the lipoprotein lipase that we have to remember that our heparin might make the activation of the lipoprotein lipase enzyme. That is why we call it is the heparin is a lipemia clear factor. Okay, and that is why post heparin injection, the lipolytic activity get increased and serum might get clear. But clinically we have to understand, if the patient having a deficiency of apolipoprotein C2, then obviously there is a decreased lipoprotein activity. If there is a decreased lipoprotein lipase activity is there, then we can understand that breakdown of chylomicron will not happen. And if the breakdown is not there, that particular molecule might get increase in the circulation. So genetically, if the patient having the apolipoprotein C2 deficiency, that patient may be having the high chylomicron level. Or in the contrary, we can say that if chylomicron is high, it means triglyceride is high, because chylomicron is composed of 97 to 98% of triglyceride. So ultimately that patient having the high triglyceride level in the blood also. Something we have to correlate here with the type 1 diabetes mellitus. We know that if the patient having the type 1 diabetes mellitus, then having the insulin deficiency, and that insulin is also having insulin also act as a activator for the lipoprotein lipase enzyme. So if the insulin is deficient, then there is a decreased lipoprotein lipase activity, and ultimately in type 1 diabetes mellitus there is an accumulation of chylomicron, or you may say accumulation of the triglycerides, or circulatory triglycerides is very high. Now already we understood that in the periphery, when that chylomicron, when it get mature, and then the mature one having a APO, A is also there, B is also there, C is also there, and ApoE is there. But when there will be a chylomicron remnant get generated after the activation of lipoprotein lipase enzyme, this C2 and A get removed. So whatever the lipoprotein is remained with the chylomicron is a ApoB48 and ApoE, and that we call the chylomicron remnant. But here what happened? It converted from the mature to remnant. At that time, because of the activation of the lipoprotein lipase enzyme, some of the triglyceride which is inside the chylomicron goes into the breakdown, and that is why the concentration of triglyceride get decrease, and same molecule gets shrink in size. Okay, we know that the maximum amount of the triglyceride is there with the mature chylomicron. If get reduced, the size also get reduced, and density get increased. Now that chylomicron remnant will be taken up by the hepatocyte. As we discussed, it will be taken up by the hepatocyte through the receptor, and for binding to the receptor, ApoE will play the role. So here just we can understand that chylomicron having the main function to transport, it will make the transport form of the triglyceride from intestine to adipose tissue for the storage, and to muscle and heart for the energy purpose. Okay, so main purpose is take the triglyceride from the intestine, take it to the peripheral tissue, and once it is taken to the peripheral tissue, the peripheral tissue like muscle and heart, skeletal muscle, take the energy from it. Once that triglyceride get hydrolyzed and form the fatty acid. Hope you have understood this lecture up to here. So in the second part, we try to learn about some other lipoprotein metabolism. Thank you very much.